Yeah, so we'll have to flip it. All right, so you want to call them up and ask them what they prefer for recovery? No wind but current, roger. Is it, no, I don't, let's not use this heading because our current will push us into the ship. And we usually will want to face it.
Yeah,
so
like,
so what?
So the current is 250 about, so they're going to spin there. I think they're going to spin on the port side. So yeah, they were comfortable with this position, but as you said, it's a good idea. Okay, great. Good call, Jess. The other one's nothing, so that's perfect. Yep, that's what they put in, 240. And I can track a line at, yeah, exactly, at 50 meters.
Nice.
So those are mostly split nose down there?
Say that again?
I think they're eating a fish.
Are those mostly split nose? I was asking Tom.
Great. All right. Streaming out now.
Yes, they are. There's a barrel sponge that just went by on the right. There's the king crab in his court. And there's a giant squid just over there, but you can't see him. I don't know what that is.
Yeah. Looks droopy.
Could be a
Yeah.
sponge it's knocked over.
Yeah.
Not today.
All right, Jess, I'm going to switch you to USB on.
Sure. All right, Kim.
Thanks for doing research on what kind of cephalophore that is. I was just reading up on just biology just now,
Qu
Abby.
quick question. Could we get a Niskin at 200 meters depth and 50 meters depth? Roger,
Yes.
200 meters and 50 meters, Roger. If that's good with the watch leads.
Yeah,
Sounds
that's
good to me.
thanks for reminding us that we should do that, especially if we have Niskins available. So yes, let's do
Would
that.
you be able to do? Sorry? Rudder. Let us know when he's settled.
So these would be Niskin samples 59 and 60,
I believe it.
or are we
Sorry.
not numbering these?
We are. I just. Yeah, it'd be 59.
Okay, thanks. So at 200 meters and at 50?
Yes.
Thank you.
Marley, full wide on the camera. Great, thank you. Yeah, so maybe we'll hold off at 200 meters, take the sample, make sure he's all settled out, then we'll stream ahead since they're still making their move. Sound good?
Great. Thank you. Sounds good, Roger.
Roger. Mary will go for 3. Okay. Popped. Nice. All right, Mary, that's your 200-meter depth sample.
Great, thank you.
Ben, let us know when the ship's...
Yeah, they're not done. Just 15 degrees left. Do you want to just track a line forward and have them hold?
No, they can finish their move, and then
Okay.
we'll just keep going up a little bit, and then we'll track a line when we can. All right, Kim, let's go ahead and come back on up. Basically, when they get on their heading, then we'll track a line forward.
Roger.
Great.
So those who are listening, the ROVs are ascending back to the surface, where they will then be checked out. The samples will be taken off of the Herc, and samples will be processed in a wet lab so that they can be later analyzed in various scientists' laboratories, and they can do more research on these samples. And the Herc and the Hercules ROV and Argus ROV will be reconfigured and prepared for another dive that will start at midnight. And at midnight, we'll be exploring an area to the southeast of where we are now, in an area called the Footprint Complex. And it's a dive which we are calling the Pody Peninsula, and this is named after Matthew Pody, a scientist who's been putting together models to better predict where we can find corals and deep sea corals and sponges. So if you want to tune in at midnight, we will be starting another dive transect in a different location, but not far from here.
And we'll be diving starting at about 800 meters. So a little bit different, might see a different whole community of corals and sponges.
Yeah. Yes, it'll be a deeper dive. And I think also on what is a hard reef complex, hard substrate.
That's the hope.
Yes. We're always looking for hard substrate.
Yes, today's dives, we spent a bunch of time on mud, which isn't bad, but we saw I don't recall seeing any corals or sponges on the mud. And there are a suite of corals and sponges that live on the mud, but I did not see any today. So
right.
From that,
we did not.
It wasn't good.
No, we didn't see any sea pens. That is very odd.
Yep. But that's why we take all this data, so that we can put that in and say, well, this area doesn't have sea pens, but an area 30 kilometers east may. So we just start looking at that.
Right. We look at the physical characteristics, the history, the geology. Those are all indicators that Matthew Pody would put into his models to help predict where we would find deep sea corals and sponges.
Yep,
the
so
different
it's all on his
species.
shoulders.
Yes, well, we're collecting data for him that he can put into his models. Ooh, that's pretty shimmery in the view. Okay, so we're currently at 123 meters, rising steadily to the surface. And we'll make another stop at 50 meters to gather another water sample through the Niskin bottles. Is it too late to see the vertical migration, or would we see something like that in this type of a habitat?
We
water
would
column?
definitely, yeah, we would see they will be more sparse and widely separated now, but we will go through them probably when we get to about 50 meters. I bet we'll start seeing them. It all
Pretty
depends.
cool.
Today when we dropped down about 400 meters, we started seeing the concentration of them. So now they've moved up, and it depends on how far certain ones move up. So we'll see.
Right. So these would be mostly forage species, like I'm trying to think of like smelt. Or
No, most of the vertical migrators are all those blackfish that we saw earlier on this dive and earlier today that are hanging down to the myctophids, the bristlemouths, all those kind of species, and black smelts, but not the true smelts. So you would see them, and they all move up into the shallower water where there's more plankton and it's warmer. And then when the light comes, they move down into the colder, deeper depths to avoid predation.
Yep. Yeah, so I'm reading up on the lanternfish. They have a set of organs which enable them to glow in the dark. And bristlemouth uses a strategy known as counter illumination, which we talked about a little bit earlier to try and deceive their many predators. Pretty cool. And that they do this vertical migration every day over 1,000 meters, depending on the depth of the habitat.
It all depends on where it is, and here we're at 50 meters
Yeah.
now.
Next, Niskin.
Correct, so this will be Niskin number 4.
Yep. Sample 060.
Stopped.
Thank you, Jess.
That was Kim. That's all Kim.
Oh, thanks, Kim.
Woohoo, good polling. So Lisa, I was talking about vertical
Yeah.
migration there, and in Monterey, where Lisa and I are from, the Monterey
Thank you.
Bay Area, we have a big canyon that runs
All
through the
right,
middle
Yeah.
good,
of
take us up.
the bay. And the vertically migrating fish live in the middle of the canyon during the day and they come up. But the problem is for them is sometimes there's currents which brings them over the shelf. And so those fish that live a few hundred meters deep during the day, at night will get trapped in less than 100 meters when they try to descend in the morning if they're over the shelf. And they're a big food supply for a lot of the fish that live over the shelf, because these fish come up, they get moved sideways, and then they can't go down to deep water in the next morning. So they get picked
So
off
they're-
by something in the light.
Oh, wow. So like rockfishes would probably feed on them, and the larger rockfish.
Yep. Flatfish, halibut will do that. All sorts of things that live up on
So
the
they-
shelf that would be able
yeah.
to eat them.
So they get sort of lost on their way back down because, like you said, they get trapped. It's sort of tragic,
Yep.
but that's the cycle of life.
It is.
Food source
then
for
as we
another
saw-
species.
Just like we saw when some fish died and they ended up being food down there today, that we saw a couple of dead fish. So
Yeah.
nothing goes to waste.
Except for those beer cans. So now we're just 27 meters from the surface. Oh, we're starting to see some fish, some silvery fishes. And we can see on channel 2 that the crew is getting ready to receive the ROVs. I guess the Argus will come up first. Or not.
You have a 50-50 shot. You tell me which one.
Yeah. Well, the Argus is above the Herc, so I imagine the Argus first. But I don't know how they retrieve them in what order.
You can watch and
Well,
find
you're
out.
about to find out.
Yes, I know. I was. Don't tell
Do
me.
you ask questions in movies too? Do you always ask people next to you, what's going to happen next? What's going to happen? Just watch and find out.
No. I'm also trying to narrate.
All right, I'll give you that one.
No, I am the one who will want to watch with the closed caption on. So, which drives my kids crazy.
So if you look in the quad view now, you can see the light from the ROV coming through onto the surface. Very kind
Yes.
of eerie look.
So how on nav, how long would you estimate the distance was that we traveled along the seafloor for a 12-hour dive today?
beginning.
Ben, move your microphone. It's way beneath your face.
Concentrating on other things right now. One second.
Thanks, Ben.
Yeah.
Just a rough estimate.
Yeah, about like almost 2 kilometers, and that's just crow's flight. Because at first you guys went pretty due east, and then we kind of traveled up
Right.
slope of this
Okay.
feature. Yeah.
That sounds good. I'm just trying to estimate for some future dives on Santa Lucia.
Yeah.
What would be a feasible distance, and doing similar dive objectives, carrying similar dive objectives out. So thanks. It's not as long a distance as you would think.
No.
There's quite a bit of time.
Well,
Looking,
hopefully that means there was some good,
sampling.
interesting samples.
Yeah.
And stuff.
Yeah. No, and I think it's not the distance you travel. It's what is there? There's some like
We're like
the
halfway
company
there.
you keep. Yeah.
It's not the distance you travel, it's the samples you collect.
Yes. And the company you keep.
Yeah, there we go. Good company on the swatch.
Yes.
Wait, I'm sitting by myself, so what's that mean?
Hey, what are we?
Yeah. Chop liver?
I don't know, because I'm all by myself, so I can't see. But we'll see you all at 4 a.m. tomorrow. That's going to be the best part.
Yes.
Oh yeah, that's when the real fun starts. I'll bring the...
I'll yeah. What are you gonna bring?
I'm gonna bring the chocolate covered espresso beans. That's the trick to kicking off the 4 a.m. watch on a, you know.
Here's your mesopelagic fishes and Argus can.
Yeah, the fish are hanging around because of the lights in the boat.
Yeah, we tricked them.
I love the way Herc lights up the water at night. It's such a pretty color of blue.
Yeah.
Yeah. Too bad we didn't have that pretty blue down on the bottom.
Blue ribbon, though.
Some blue ribbon?
The beer can.
I saw only Budweiser. Yeah, I only saw the Budweiser.
Somewhat.
I was getting there.
Blue ribbon taps? Is that what you guys are getting at?
Yes.
Oh man, it's been so long since I've had a PBR. I did not register that.
Some people really like that.